Ever wonder what makes you you? The answer lies within your DNA. This is genome mapping and how it's transforming our understanding of health and disease. What is genome mapping? Imagine a vast library with millions of books, each containing a single chapter of a giant story. That's kind of what our genome is like. Genome mapping is the process of identifying and arranging all the genes in their proper order on a chromosome. There are two ways of doing genome mapping. First is genetic mapping, and the other is physical mapping. Genetic mapping is like that reunion of genes. It doesn't tell you the exact order of all the genes, but it reveals which genes are close together on a chromosome. We start by collecting DNA samples from a family with a particular condition or trait. The DNA is then isolated from the cells in the sample. Then we examine the DNA closely to find differences between people with and without the trait. These differences are like sign spots that are called markers. By following these markers across generations, we can track how the trait is inherited. If a marker and a gene are close together on a chromosome, they're most likely to be inherited together. The further apart a marker and a gene are, the less likely they are to be inherited together. The more markers we have, the more detailed genetic map will be. While genetic maps are a good starting point, they need additional information from other techniques for pinpointing genes precisely. There are two main ways to do genetic mapping. First is by family studies, where scientists track how traits like eye color or disease, which are inherited across generations in a family. By looking at the patterns of inheritance, they can infer which genes might be responsible and how close they are located on a chromosome. Second is by crossbreeding experiments, where they breed organisms with different traits and observe how those traits are passed on to offspring. This allows them to map the relative location of genes that can control these traits. Genetic mapping gives us a relative idea of gene locations, but what if we want the complete picture? That's where physical mapping comes in.
To know the exact location of the genes on the genome, the physical mapping is used. So let's discuss what is the physical mapping. Imagine you have this giant book ripped into the millions of the teeny pieces, all jumbled up. Physical mapping is like figuring out the order in which those pieces go to reconstruct the entire book. Here how physical mapping works. Scientists use special enzymes to cut the DNA into the smaller fragments. Then these fragments are analyzed to identify the unique sequences or landmarks within them. Techniques like overlapping regions and the landmark locations help scientists piece together the fragments in the correct order along the chromosome. So let's discuss what are the techniques that comes under the physical mapping. First of all, the fingerprint mapping. The physical map constructed from fingerprinted clones have been widely used in the genomic research for both genome-wide and the region-specific analysis. In fingerprint mapping, the genome is broken into the fragments. It is denatured, which are copied into the bacterial cells. The DNA copies are then cut by the restriction enzymes, and the lengths of the resulting fragments are estimated using a lab method called electrophoresis, which separates the fragments of the DNA according to the size, resulting in the distinct banding patterns. The fingerprint map is constructed by comparing the patterns from all the fragments of the DNA to find the areas of the similarity. Those with similar patterns are then grouped together to form a map. Fingerprint mapping was used to form the basis of the human, mouse, zebrafish, and pig genome sequences. Coming towards the optical mapping, a technique for constructing ordered genome wide, high resolution restriction maps from single stained molecules of the DNA called the optical maps. First of all, the cells are lysed to retrieve the genomic DNA. Then the single genomic DNA molecules are placed onto the microfluidic device. The restriction enzymes are added to cut the DNA molecules at specific positions. Then each DNA molecule is stained with a fluorescent dye. A optical map of single molecule are derived by measuring the fluorescent intensity. The overlapping of the multiple single molecule maps gives us the consensus sequences, the genomic optical map. Just like this, one more interesting technique is FISH, fluorescent in situ hybridization mapping. The FISH technique works like searching for a specific address on a giant chromosome map. First, we prepare the cells and extract the chromosomes. Then, we design a fluorescent probe, a tiny messenger with a specific DNA address. This probe is introduced to the chromosomes and searches for its perfect match. Finally, we use the fluorescence microscope to see where the probe lights up, revealing the location of the target DNA on the chromosome. So, FISH mapping uses fluorescent probes to detect the location of the DNA sequences on the chromosomes. These probes can be directly or indirectly binded to the fluorophore using haptens. Firstly, the probes are designed. These are short sequences of the single stranded DNA that match the DNA sequence that the scientists want to find. The probes are then labeled with the fluorescent dye before being mixed with the chromosome DNA so that it can bind to the complementary strand of the DNA on the chromosome. The fluorescent tags allows the scientists to see the location of the DNA sequence on the chromosome. Last but not the least, the sequence tag site STS mapping. This technique maps the position of the short DNA sequences 200 to 500 bases in length that are easily recognizable and only occur once in the genome. These short DNA sequences are called sequence tagged sites. To map a set of STs, a collection of overlapping DNA fragments from a single chromosome or the entire genome is required. First of all, the genome is broken up into the fragments. It is denatured before being replicated up to 10 times in bacterial cells to create a library of the DNA clones. The polymerase chain reaction (PCR) is then used to determine which fragments contain STS. Special primers are designed to bind either side of the STS to ensure that only that part of the DNA is copied. If two DNA fragments are found to contain the same STS, then they must represent overlapping parts of the genome. If one DNA fragment contains two different STS, then those two STS must be near each other in the genome.
In 1990, an ambitious international effort called the Human Genome Project began. The goal was to map the entire human genome for the first time. It was a monumental task taking 13 years to complete. In the human genome mapping, physical mapping techniques was used, where fragments with overlapping sequences was used to establish their order. Using known DNA sequences as anchors to position unplaced fragments, we generated the physical map, which wasn't a literal map, but a digital representation showing the order of DNA fragments along the chromosome. By identifying genes linked to specific diseases like cystic fibrosis or cancer, scientists can develop better diagnostic tools and treatment strategies. Companies like Illumina and PacBio are developing advanced sequencing technologies to facilitate this. One day, doctors may use our individual genome map to tailor treatments and preventive measures to our specific needs. Companies like Helix and 23andMe are at the forefront of exploring personalized medicines based on individual genomes.